Hello, I'm Haruo Suzuki from Keio University, Japan. Thank you for your introduction, and thank you for organizer for giving me this opportunity to talk about our research. Okay, today I will be talking about plasmid host range and the nucleotide composition. First, I'm going to talk about the background introduction and discuss determining plasmid host range. Then, I will show you some result of comparing plasmid and their host chromosome regarding nucleotide composition. After that, I will show you result of predicting evolutionary host range of plasmid based on nucleotide composition. Finally, I show you result of comparing transconjugates, nucleotide composition, and other bacteria. Okay, this is a genome structure of typical INC-PM plasmid. So INC-PM plasmid genome can be divided into two parts, backbone gene region and accessory gene region. Backbone region consists of essential plasmid function like replication, plasmid maintenance, stability, and conjugative transfer. The accessory region encodes host beneficial traits like virulence factor and antimicrobial resistance. So there are many INC-PM plasmid reported so far which carry antimicrobial resistance genes in their, for example, Neisseria gonorrhea encoding tetracycline resistance, and INC-PM plasmid in Klebsiella encoding colicin resistance genes, MCR1. So it is important to predict plasmid host range, meaning range of hosts in which plasmid can replicate or transfer. So this slide shows the plasmid transfer from a bacteria to another bacteria. So if plasmid transfer, this is a big problem because they can spread antimicrobial resistance between different bacteria in environmental microbial community. So in this study, we focus on predicting plasmid host range and check their experimentally determine experimental transconjugates. And I show here three key points why we should care about plasmid host range. So first, ecological question, so gene flow among bacteria. Second is medical interest in reservoir of antimicrobial resistance and virulence factor plasmid. And finally, it can be used for application, so we can search, choose for cloning vector for specific bacteria. Okay, next. Determining plasmid host range. Plasmid host range can be usually expressed qualitatively, narrow host range plasmid and broad host range plasmid. And plasmid host range can be determined two ways. Laboratory mating assay and also plasmid associated taxa, meaning so we can predict range of host of plasmid based on the taxa in which plasmid have been found so far. Okay, next. So in our study, we focused on nucleotide composition to predict plasmid host range. So first, I show you result of nucleotide composition similarity between plasmid and host chromosomes. This figure shows similarity in nucleotide composition between plasmid and host. So y-axis indicates plasmid GC content. X-axis indicated their non-host chromosome GC content. So you can see, so very strong positive linear correlation. So plasmid chromosome GC content are very similar. And we also investigated oligonucleotide composition like dimer, trimer, tetramer. This is an example of trimer, threemer. So this sequence, example sequence, ATGG, consists of two trimer, ATG and TGG. So we compared these nucleotide composition trimer between narrow host range and broad host range plasmid. And we found that narrow host range plasmid tend to very similar to their host chromosome. And next, we predict evolutionary host range of plasmid. So this nucleotide composition can be used for predicting plasmid host range because plasmid tend to be similar to oligonucleotide composition to their non-host. So this means plasmid have acquired, gained the host nucleotide composition over evolutionary time. So we can apply this for predicting different plasmid host range. And I show here different results, but maybe I skip here. Okay, this figure shows number of candidate evolutionary hosts for each of the plasmid based on trinucleotide composition. So the bar indicates the number of hosts in which plasmid have been evolved. The plasmid above the line are narrow host range plasmid like INC-F, H, I plasmid. The plasmid below the line indicates broad host range plasmid like INC-N, P, W. And the different color indicates different taxonomic groups, alpha, beta, and gamma proteobacteria. Narrow host range plasmid like INC-F, F plasmid show the very blue. So only gamma proteobacteria are candidate host range based on nucleotide composition. In contrast, broad host range plasmid below the line show very colorful, right? So their evolution host range belong to different class, alpha, beta, and gamma proteobacteria. And we, excuse me, we also quantified the diversity level of these candidate hosts based on 16S ribosomal RNA gene alignment. And the result shows INC-PM plasmid show higher host diversity level than INC-F and other narrow host range plasmid. Okay, finally, I will show you result of comparing experimentally obtained transconjugates and other bacteria. So this experiment was done in the Dr. Shintani's laboratory at Shizuoka University, my collaborator. And on the left, show the donor cells containing 4 different plasmid, GFT tagged plasmid. And the recipient, as a recipient, use environmental samples like soil or water as microbial community as a recipient. And then conduct mating assay, filter mating assay. Then plasmid transfer code. And after that, transconjugates, meaning bacterial cells with plasmid, can be sorted by GFP expression. So they sorted at single cell levels. Then performed sequencing to determine DNA sequence of transconjugates. This box plot shows distance in Mahalanobis distance between plasmid and bacterial chromosome. So colored box plot indicates similarity, dissimilarity in nucleotide composition between plasmid and transconjugates. The gray box plot indicates Mahalanobis distance between plasmid and non-host chromosomes. So as clearly shown here, similarity in nucleotide composition between transconjugates and plasmid are highest. So lower Mahalanobis distance meaning highest similarity. So this result shows that, as shown, described here, nucleotide composition of plasmid were similar to those of the chromosome of the experimentally determined transconjugates. Okay, this is a summary take-home message. So our goal is to predict plasmid host range we compared plasmid and chromosome nucleotide composition, and we found the following three results. Plasmid resemble non-host chromosomes. Second, broad host range plasmid resemble a broader range of bacteria than narrow host range plasmid. And finally, we demonstrated that plasmid resemble transconjugates compared to other bacterial chromosomes. Okay, so let me announce the International Plasmid Meeting, which will be held in Hamamatsu City in Japan on September 2nd to 6th, 2024. Please save the date. And I would like to meet you at the meeting so we can share information on antibiotic resistance and their vector like plasmid and phages. So yeah, I would like to thank Otsumi Frontier Science Foundation and would like to thank collaborator Dr. Hirokazu Yano, Masato Suzuki at the National Institute of Infectious Disease in Japan, and also Dr. Masuki Shintani at Shizuoka University, and Dr. Hideaki Nojiri at the University of Tokyo. Thank you very much. Thank you.